Hello, everyone. Welcome to Foresight's Biotech Group. Just a little bit of housekeeping before we get started. There is a Vision Weekend conference at the beginning of December, the first weekend of December. It's our main in-person gathering, and we'd love it if you joined us. With that being said, we're really lucky to have Wesley Clawson with us today. He's in Michael Levin's lab, and he has a background in electrical engineering and neuro. And today, he'll be talking about training tissue. So with that being said, Wesley, feel free to take it away.
Thanks. Let me share my screen real quick. Cool. Well, hi, everyone who's there. So my name's Wes. That's not a lie. I'm here to talk about what I do within the group of Mike Levin. And the title is a little bit different than what I proposed, but it's close enough. And my intro is a little bit different than I planned because I think Lydia sent me a question that I was supposed to answer ahead of time and I didn't. It's what do I want? What could be solved in my field that would be great or make my life better? I can't remember the exact wording. So I thought I'd give an example first. So maybe it's a different intro because I'm presenting the problem first. And it comes from a funny show called Taskmaster, if you've ever heard of it. It's a comedy show where there are tasks and people try to do them, and whoever does the best wins. And the prizes are pretty nonsensical. And one of the tasks that I think is pretty funny was like camouflage yourself. So they were going to take a photo, you had to camouflage yourself and hope for the best. And here is what the contestants did. So there was 5 of them, and you could probably scout for a minute and take a guess. I'll leave it up for a hot second. So there's a person in each photo for sure, but the one in the bottom right I think is the most obvious. But then I'll reveal where everyone is, so maybe take a look. It's not super important, but notice that every photo is a little bit different. So one contestant literally took the door off and hid behind it. One contestant, if you're not familiar with the show, is number 9. So he hid himself by hiding among kind of like people. One woman got under a tarp or a piece of turf. Someone hid under the flowers. And then this guy just laid down, took a photo of himself looking like a banana, and then put it on the fruit stand. So here they are again. So through the door, someone hiding, the banana man, someone in the dirt, and someone behind the flowers. And this has almost no biological relevance other than the word camouflage, but the idea is that they all have roughly the same physical form. Okay, they all have a body with 2 legs and 2 arms. They are all roughly the same height. Nothing distinguishes them greatly they are the same species. Yet when given a goal, they did them all in separate ways. So really the big difference, if you ask, like, what's the difference between these 5 contestants, because they're also all comedians, is like their personality, like who they are. So they came up with a different solution. This idea of coming up with multiple solutions to the same problem is the challenge that I would like to see solved for progress in my field, please. And I think this image shows it quite well. So these are the three pieces of this. The example that I just showed is called Degeneracy, where if I have a problem or something that I'd like to do, like write a sentence, there are multiple ways to do so. I could use a pen, crayon, or a pencil. Redundancy, we're typically familiar with in biology. If I need to write a sentence, I'd like to use a pencil, and if one breaks, I have 2 spares, and that's fine. So typically we're familiar with redundancy. We're also somewhat familiar with pluripotentiality. With a pencil, I can do many things. This one's more familiar because of pluripotent stem cells. It's a cell that, given different opportunities, can do many things. But degeneracy is the trickiest one, in my opinion, and one challenge that that drives a lot of my work, and I don't have an answer. But if someone could figure out how to meaningfully track and investigate degeneracy in a system in a meaningful way experimentally, I would love to hear your thoughts. And when I go to 2 ends of a scale that are important to me, here's one from Yves Marder and Gouillard. So it was published in 2006, where there was 2 behaviors that something could exhibit: the purple behavior and the orange behavior. This was done in a model, and those behaviors are governed by some kind of mathematical equation that have parameters inside of them. So as you can see here, there are many different sets of parameter A, B, and C that produces the purple behavior, and then same thing for the orange behavior. Many different ways to get orange. Okay, so you would say these are all degenerate solutions to the same thing. I'd like purple, or I would like orange, and then here are all the different comedians hiding in the different way. They still achieve the thing, but differently. The problem, in my opinion, gets worse and worse the higher the scale up that you go. For example, in epilepsy, there are many reasons someone can have epilepsy, or when you have epilepsy, many pieces of the scale that can be where this epilepsy maybe propagates from, such as the genes, cell signaling, channels, networks, whatever. And at the end of the day, all of these different things, all these different etiologies will lead eventually to cognitive deficits and seizures. And it's what makes epilepsy a very difficult disease. Is the one marker we have is that you have seizures, and they can come from a variety of places. So not only can degeneracy be good in a sense that I can solve the problem in many fun, unique ways, but also problems can be generated in a lot of fun and unique ways. So if we're trying to help someone with epilepsy, it's very difficult. And I mentioned scale. This is a really nice figure from Stuber. This degeneracy, of course, exists across scales. Ion channels exhibit degenerate behaviors. There can be degenerate morphological properties, synaptic properties, all the way up to system-level interactions. So again, you're seeing behaviors, and they can be created by a multitude of different things underneath. For example, all cars work like a car, but how you build them can be quite different. Of course, they have different properties, but under the hood, an engine is an engine, plus or minus. And what's really interesting is that if you're trying to work towards curing a disease or making a disease state better, there are many different ways perturbations can change things. For example, here in a parameter space, some cartoonish parameter space, many different types of perturbations can lead to the same kind of functional outcome. So there are many different ways for a healthy person to become pathological. So one, it's tricky to diagnose and figure out where it came from, but then if your job is to fix this, it gets very difficult. For example, here's a graph of anti-seizure medication starting out from the 1850s all the way up till almost now. So this was published just a couple of years ago. And the number of drugs is increasing. If you're unfamiliar with graphs, it goes up because there are so many. But a fun fact, Gallers back in 1881 was noticing that they only had one anti-epileptic drug called bromide at the time, and the percentage of unimproved cases went under this treatment was about 30%. Okay. And then in 2021, Sultana did a nice survey of a survey of many different epilepsy studies, and drug-resistant epilepsy remains at roughly 25 to 30%. Now, for sure, the way that we investigate and diagnose epilepsy has changed, so the number of cases can go up. But the stark truth remains that despite lots of work, imagine all of the FDA things that went into all of these, we basically made negligible progress because of this degenerate issue. So in the same way that they hit in different ways, epilepsy can affect you in different ways, and drugs can only do so good. Now, I'm going to talk about degeneracy more in a fun, useful way while keeping the epilepsy example in my mind. Most of the time, if you see an animal in some kind of environment in a maze, when we see an animal solve the maze, or when you see a dog learn to sit, you think, wow, that's incredibly smart. This is very cool. And the point of the talk today a little bit is to help you shift your brain. And this is the Foresight Institute, so you probably don't need any help at all, and I'm probably behind you. Is that I think you could say this about any biological system in an environment when there's a goal or something that they would like to achieve, like camouflage yourself. When they solve it, you should think, wow, that thing is smart. Look at it, do the thing. And because whether you're an animal or a biological system, I think each has an unveil. And this is a word coined far in the past, which is like constituted by more or less a broad series of elements called carries or significance or marks, which are the only thing that are of interest to the animal. So in the past, everyone cared about animals because we were trying to figure out how the hell they came here. And how did they get to the way they were? But I think you could easily cross out the word animal, put in system, and keep the same thing. So no matter the scale, no matter the biological system, there will be markers of significance, these environmental variables that are of great importance. And typically, if you're going to pursue a goal, a task, or something in this environment, those are the things that matter. So when you do experiments, you should always imagine yourself a bit as the system. And again, I think the coolest thing that biology does is in these environments, they will learn. They will adapt, learn, respond. Whether that's goal-directed or homeostatic, to me, it doesn't super matter. They learn to continue to exist. And like we mentioned, this can occur across scales from genetic regulatory networks, neural networks, tissues, animals, people, groups, societies, whatever. It occurs across scales, but as well as spaces. So for example, we're very familiar with 3D space because we exist in it. But for a genetic regulatory network, if we're talking about the unveil, these carriers of significance, you might be thinking about transcriptional spaces. Or if I'm an animal developing, I'm thinking about morphospace. Or if I'm something with epilepsy, maybe I'm thinking about this physiological space of potassium and pH. Who knows? And something that, I mean, I would credit this far more to Mike and people before me, thinking about intelligence is kind of the degree of competency that any system has in navigating one of these spaces. And this is where degeneracy and intelligence kind of start to interact for me, because there's no wrong way to eat a Reese's. If I have some kind of goal, if I would like to navigate a space, there are many ways to do so. Sure, they may be local optimums, but if you can overcome certain barriers and find better solutions, the better and more you can do this, I would argue, the more and more intelligent we could call you. If you're bound only by gravity, for example, not very intelligent, there's nothing you can really do. But as systems get more intelligent, they can find better and better solutions. And there's evidence for this, again, across a number of spaces. For example, down in transcriptional space, in physiological space, I mentioned this with the Martyrs group, and then an example from Mike's lab is that if you take a planaria and you put it in water with some barium, which it has never experienced in the past, its head will degenerate, so it will explode. And if you don't know anything about planaria, they are infinitely regenerative in the correct conditions. So if you put in barium, not a great condition, it has never seen barium in the past, its head will degenerate. But then after time, its literal morphology changes, it will regrow a head that is barium resistant. So this adaptation, this response, this navigating kind of a genetic and morphological space in order to overcome or achieve this goal of staying alive. I have done some work in transcriptional space with Saurabh Biswas, if you're interested in this, where we approach this problem of degeneracy and trying to solve spaces in the realm of computational models of things. It's pretty cool if you want to check it out. And to give a little bit more insight into this model that I talked about before, where there's two behaviors, 1 and 2, you have degenerate solutions to them, but you can see the behaviors. Eve Martyr happens to do it in the lobster. 1, because they're delicious, but 2, they're very interesting. They have this circuit inside of them that helps work their stomach and breathing, most important, their stomach. And these are the neurons that constitute that circuit and roughly how they're wired. And if you look at 2 behaviors that this circuit can demonstrate, the pink one and the blue one, if I asked you to find the difference between those 2 behaviors, you would be hard-pressed to find anything radical. Of course, they are slightly different here and there, but on the whole, they are the same. And what's absolutely wild is that if we look at the membrane conductances, which help determine the electrical properties of cells, as well as the synaptic conductances, which is how they communicate, here you can see the parameter space for the purple and the blue circuits, and they are radically different. So again, a computational evidence that these degenerate solutions exist. And then Eve's group also looked at biological relevance, how they resist to perturbation. And even in the most conservative cases, there are orders of magnitude, thousands and thousands of solutions that all fall within the bounds of reality, that all respond well to perturbations and could very likely exist. And they've been experimentally testing many of these. Okay, so we have this idea of degenerate solutions. Great. We have these goals, and biology seems to do stuff, in order to solve these things. The way that I think about how they do this is through something called computation. And I want to just briefly go over it because it kind of highlights how I think typically. If you would like to do a thing, have a goal, for example, have some pyloric rhythm in the lobster, or fly if you are a bird, of course you need wings, or in the lobster, of course you need neurons with synapses. However, if you just think about the thing they are trying to do, as well as what they have in order to achieve it, you may miss this kind of how. Like, what are they actually doing? And in a cartoon, the bird needs to flap. And typically in neuroscience, what people do is say we have neurons, they spike and stuff, and then we have cognition, and we hope for the best. However, I think I mean, one, you have to pick a level of investigation. We can't investigate every level of biology in a single lab. But here, this is kind of constraining the idea to this kind of whole organism. Somehow neurons spike and support cognitive demand. I'm not saying that they don't. I'm just saying that the viewpoint sometimes is a little limiting, especially because if we think about this idea that any biological system could be intelligent, navigating spaces towards goals, it gets very tricky. Okay, so basically, I've built a talk setting up that biology is very complex and very hard, and I don't really know how to solve it. But I've built a research program that's trying to get at one tiny facet of it. And that's looking at the how and the what of a biological system and how they pursue these whys. Built on the fact that biology can learn, happens across scales and spaces. They are intelligent and can find degenerate solutions to these problems, and it exists across scales. So any work that I think should be done towards this needs to exist across at least two so that you can, talk about them meaningfully. So to bring it back to this picture that I showed, I think redundancy, degeneracy, and pluripotentiality are kind of mechanisms of problem solving or intelligence. And so a lot of what I've started to do and plan to do in the future is rely on looking for these mechanisms every chance that I get. Because redundancy, degeneracy, and pluripotentiality can look very different depending on the system that you're investigating. Mike and I had this crazy idea to discuss what this would look like in the future if you could do all these things in this nice paper that I think is worth at least skimming, is that these fundamental principles governing how biology works, how they solve these problems, are super obscured. Hopefully, I've made the point at least somewhat clear that because of the scales, because of the degeneracy, because of the redundancy, it's very difficult. It's evolved and complex. And so what we decided to do, Mike led this effort, is to propose this kind of goal-seeking, this teleonomic goal-seeking problem-solving behavior exists regardless of what type of being you are. And if you take a program such as this, if you go through and do as many experiments as you can of living forms, one that as exist, but two, especially as never existed before, could help you understand, one, the design principles that we see, but also the design principles of life as it can be. We're quite familiar at this point with microbes, plants, sort of animals, no clue how the human thing works. But as we get into these design machines, artificial intelligence, this kind of beautiful web of life as it can be is very investigatable. And to do this, the reason I kind of went to Mike's lab to begin with is because of this TAME paper that he wrote, where he asked if we think that things are intelligent, or that means maybe we can persuade them or train them. For example, I think everyone has seen a dog in their life for the most part. I have trained my dog quite well. When I go like this, she sits, and I have no idea how a dog works, but I know that with the right training signals, I can drive her system in order to sit when I do this. She is very trainable because I think dogs are decently intelligent. If I do this to a rock, it doesn't matter how many times I do it, I probably won't see anything. So if you're going to investigate a system, you need to pick kind of where you think this system sits on this axis of persuadability. Humans, I can talk to you all day and probably can convince you of a couple things. Dogs, I can get to even, Pavlov got them to drool in a radically amount of weird situations. Or is it something like a homeostat where there's just a basic set point and the system balances? Or is it something as unpersuadable as a clock where in order to change its behavior, you have to not only have an enormous amount of mechanistic knowledge, but you have to have an enormous amount of effort in order to do so. Talking to you today, as an inverse example is quite easy. And I have no idea how any of you work, but I might be able to convince you that what I'm doing is like at least partially okay. And in my work, I'm going to start from C. Okay, so if we think there's intelligence, if we think they can solve problem sets interestingly, what if we could train an artificial goal, like a dog sitting, observe the what and the how very closely during and after training to see if we can uncover, one, how they do it, and if we can find things like degeneracy, pluripotentiality, and redundancy. I have not fully answered that question, but we started. So if we want to be able to set an artificial goal or a biological system, I do so through something called embodiment. I did not invent this, of course, because In this type of framework, you need an environment that can act on an organism, some kind of mind that at least partially processes it. So it needs to have markers of significance. The environment needs to be those. And then a body changes your relationship to the environment, whether you couple with it or move away from a signal. So we virtualize the environment in the body. And we're not the first people to do this. And I'll go over some examples in a second. But it looks like this, where we're going to grow tissues. And today I'll talk about neural tissues in a specialized chamber that lets us look at them. Whether, today we'll talk about their electrical measurements called ephys or electrophysiology. The environment will be running on a computer, and the actions that the culture takes will influence what goes on in this virtual environment. And based on the rules that we've decided, we can apply punishments and rewards. And then hopefully train it in the same way that you would train a dog. To do so, we use CMOS electrode arrays. They're very fancy electrodes that are very tiny. In the past, electrodes were big and fat, and so it was very hard to record many neurons at once and know what they were doing. Here, the electrodes are very tiny. As you can see here, there are these purple things. And so you have kind of a fun opposite problem where instead of having many neurons to one electrode, you have many electrodes to one neuron. And on the right is the data that you get out, at least a degree of it. So if a neuron spikes, we can pick it up and mark it with a dot. So here on the y-axis is all the channels of my electrode. There are 1,000 in this example, and I record it for 5 minutes. And every time there was a spiking event on the channel, I mark it with a blue dot. If I let all those blue dots kind of fall down and look at the average activity, this is what it looks like. There's some quiescent periods, and then there are these large bursts of activity where many of the neurons synchronize and are firing simultaneously. They quiet back down, they burst, they quiet back down. There are theories of why they do this. None of them are extremely validated. It's still an open topic of discussion. And if it's something that someone wants to talk about, I'm happy to. So this is pretty cool. When neurons fire, there are fields. These fields collide, sometimes so much so that we think bursts happen. But because these electrodes are so small, we can do things like this, where when there's a neural spike that's in red, we can actually watch it travel along the axon. We have to reconstruct these after the fact, so to do this in entirety in real time is currently impossible. But here you're watching a spike fire, and then which takes about, a short amount of time, and then it travels along its axons. And the red lines here are tracking roughly where we saw it. And if you see the line, that means we're decently confident that it's there. So we can build these nice, pretty maps of the neurons in my dish. And we can get a lot of properties out about them, like what was their firing rate, how fast did the action potential travel along the axon, how far did it go, how many times did it branch, etc. Of course, I don't do this alone. That would be insane. To grow these things, Juanita taught me how to do this, and now Danielle and Sydney work on my team. And we take mouse or rat cortex. You take them from animals just before they are born, before the neurons really kind of establish themselves. And there's a way to get them from a brain onto a plate. And this is what they look like. They grow on these nice big networks. So kind of like a neural network, if you're familiar with those, but, in real life. And again, we want to take some of their behavior. Here today, I'm going to talk about this bursting behavior that's quite easy to see because it's the whole plate being like the wave in a stadium, if you've ever seen that when people stand up. It's slow-ish relative to, us. It's easy to see because it's across the whole dish, and it's something that the whole culture has to do together. You can't have 1% of the tissue burst. A burst by definition is this culture-wide activity. And so the results I'm going to talk a little bit about today are about we are going to try to train around this burst behavior. And I'd like to point out I'm not the first person to try to embody neurons or to train them or to interact with them in a meaningful way. There's been many others. Most famously, the Shahaf and Marom were the first ones to do it. And then Lana Senapayan did some excellent work trying to guess at why this works, called stimulation avoidance. I'll talk about it in a little bit. Urs Frey had them control a robot a bit. Steve Potter, I think, is probably the most famous. He did a ton of work down in Georgia Tech. And then recently, a private group called Cortical Labs tried to get neurons to play Pong. And it worked pretty okay. Again, here's the people that helped me do this next part. Nathan, who's at Harvard, Mirage, who I think is still at UCLA, Evan, who's now in Florida, and Trevor, who was stolen by Google, and Caitlin Grasso, who works with me here at Tufts. So they helped me build this virtual embodiment paradigm. And the idea is we wanted to treat the tissue as a whole. We believe that it's intelligence, it can make choices, it can do things, so it's a bit agential, and we built it in order to do learning. And I like stupid names, so we called it the Holistic Agential Learning Platform. Holistic, we're going to train off bursts. Agential, because all the tiers inside can make intelligent decisions, and again, we're doing learning. So when we see these bursts, I have a lot of GIFs, so it makes my point more clear. Sometimes they start on one side of the chip. For example, it started on the right and bursted to the left. So I'd call this like a leftwards burst or a burst on the right, which is a bit more straightforward. Sometimes they start on the left and go to the right. Killer. It's the simplest behavior ever. They do it every once in a while, and it's very easy in real time to see if it started on one side or the other. If we wanted to embody it, here they kind of have this virtual body they have, the actions that they take to affect the environment. There's only two choices. I'm going to burst from the left or I'm going to burst from the right. And then we have to decide, okay, if we would like to train to only see bursts from the left, for example, we need to have some kind of system of punishment and rewards in order to see change. So we have two paradigms. One was based off, I'll talk about this actually bottom one first, which was built off the ideas of Shahaf and Sinapian, who I mentioned earlier, where, whoops, I flipped these around. This should say stay random stim, and this one should say no stim. Oh, my labels are all messed up. Hold on. This is important, so let's change them. Okay, there we go. Great. Now the figure makes sense. Okay, so let's say you do a behavior, and if it's something that I don't like, I blast you a little bit with electrical pulses. And you could imagine that if you're doing something wrong and someone shocks you a little bit, somewhat randomly, it's annoying and you would like it to go away. But then when you do some kind of behavior, the stimulation goes away. And you're like, okay, well, I'll just do that. So imagine if every time you sat in your chair, I shocked you. Eventually, you would try everything. You'd start like touching objects to see what happened. And then the moment you stand up, it stops. And you're like, oh, great. You sit back down, it shocks you, you stand back up, and you're like, okay, it's the chair. And then you move on. This is the basics of stimulation avoidance. There's something you don't like, and you will search your action space in order to make that go away. So we use this paradigm mostly. And we've been trying recently to build a paradigm based off of Carl Friston's idea of the free energy principle, which was also used in this Pong paper, where the general theory is that biology hates when things are random because they're unpredictable. It's hard to learn from them because I can't really tell what's going on. I can't integrate it into my system. It's very hard to predict and make decisions. So under this paradigm, when they do something we do not like, we will randomly stimulate them at a certain frequency. And then when they do the thing we like, the stimulation will become regular and very predictable. And the idea is that if FEP truly is something driving biological behavior, this should train similarly to something like stimulation avoidance, which we know have at least more experimental evidence that it works. Cool. Okay, so we're going to watch bursts. I want to train them to go from one side to the other. And honestly, it doesn't matter which side. I'm going to say this side I trained for and this side I trained against. And we're going to have some kind of positive and negative feedback depending on what we do. Boom. Okay, some data. Let's look at it. So here, every line is a culture. And when we see a dot, that represents that I did a recording for a bit. I trained them under one of these principles for 20 minutes, and then I recorded them for a bit longer. And I'm trying to look at the proportion of correct bursts, meaning on the correct side, as a function of days or training. So these top two graphs were both done under the stimulation avoidance paradigm, where to reward the culture for bursting correctly, I turn off the stimulators. And the stimulators are uniformly distributed across the tissue. I'm not stimulating a specific side. I'm not doing anything tricky about trying to hit a specific neuron. I just randomly stick the stimulators down. And so this neural tissue, you see in the beginning, has kind of a distribution of the side that it tends to burst from in the beginning. And over days, on average, they all go up. So the more that I train them, the more the behavior that we want goes up. So somehow this neural tissue in the stimulation avoidance case, that there is a harsh stimulation because it's rather strong based on a behavior. It is somehow searching its action space. And here I would roughly approximate over days or something, or it searches it within the training period, but then has to integrate this knowledge over days. And eventually, almost all of these lines go up. Here, this is the second round with a different group of cultures. And you see in the beginning, some of them actually do opposite of what we want, but then after some time, ramp back up again. Of course, this is a relatively low N. These are somewhat not hot off the press, but recent. So we think through stimulation avoidance, we're able to train neural tissue to burst from a specific side of the dish. Meaning they have to utilize these degenerate, redundant, pluripotent mechanisms of problem solving to reorganize synapses, firing something in order to solve this goal state that I've given them. We've been trying to run this FEP paradigm where to reward them, we just switch the stimulator from a regular pulse to a random pulse to a regular pulse. And in one case, that worked quite well, but in other cases, it did not. There's, of course, a lot of tuning to be done, but the jury is still out on how well this FEP-based feedback works, at least in our system under the parameter set that we chose. And then, because we're not terrible scientists, here are some controls where two of these cultures, and again, every line is a culture, two of them were stimulated under a different experimental paradigm, has nothing to do with bursting on one side or the other. And then 4 were never stimulated ever. And this proportion never really shifts. So we think that really it's not, these lines aren't due to some developmental factor over days. We really think it's because we are, doing some kind of training paradigm. Cool. So to kind of, rather than bombard you with more and more graphs about what I think is happening, I think it's fun to go over the behavior changes and then to think a little bit. So to put everything together, I think these kind of mechanisms of problem solving, of redundancy, degeneration, and degeneracy and pluripotentiality are really important. And if I can make this pretty cartoon very ugly by saying what we've been trying to do is in the future, because again, I'm still somewhat of a young investigator, meaning my career is early, not because I'm physically that young, but we can change different whats, train them for a fixed Y, and see if you change the neurons, if you change what system you're growing in the dish, how are they achieving a similar goal? We can train a goal across the same neurons and see all the different, ways that they might solve it. For pluripotentiality, what if we train 3 different orthogonal goals for the same dish? Do they use the same mechanisms or not? So the challenge that I would like to see solved is the challenge that I'm currently working on, is due to the high intelligence of biological systems, meaning there are many different ways they tend to solve problems. Can we sit down and empirically test this experimentally in the lab in order to understand not just, oh, for this one neuron, this one synapse changes, this one gene goes up or down, but on the whole, can we find kind of design principles that are embedded across multiple different types of systems using this closed loop feedback system? So some of the current and future work, sorry to leave it a little vague, is that maybe can we train goals to change over time rather than a static goal? Can we train them to ask for media changes so their behavior gets coupled to some kind of chemical stimulation? Speaking of the epilepsy case, if I can change bursting location, can I train epileptic tissue to suppress epileptic regions of the tissue? As a fun joke, can we get them to play Doom? I mean, you can run Doom on anything at this point, so it'd be very fun to run Doom on neurons. And then, of course, we have data on some of this, but I didn't want to bombard with graphs, is be able to characterize cells, tissues, whatever you're looking at throughout these learning processes. So transcription factors, synaptic markers, tracking these axons, how are the sheathings working? I mean, there's unfortunately a high degree of things that neural tissues have at their disposal in order to change. So we have to go through and start looking at as many of them as we can at the scale that we've chosen. In the future, integrating different tissue types, kidney, heart, iPSC cells, start to do optogenetics, use microscopes to look in a little bit better in real time. We have a variety of tasks and timescales that we can train cultures to work towards, with the end goal of kind of exploring the relationship, even if virtual, between this like mind and body, searching for what the heck is all these biological systems doing in order to stay so competent. And if we can find those things, can we then put them in our engineering, into our computers, into our AI, or to abuse these things in order to make people's lives better through, for example, training away disease states? Cool. With that, I just want to say thanks. The current team members that are responsible for me being able to do anything: Caitlin, Sydney, and Danielle. So my past team members, 11 lab members that have helped me in some form or another, the 11 lab admin team, as well as my funders. If one of you happens to be, just some hyper wealthy person that loves my work, your name could also go here too. I can hang out to answer questions, but you can also feel free to email me here. Thanks.
Thank you so much, Wesley.
Yeah.
I think we can open the floor for questions. I know that there was a couple from Steve in the chat. So if you'd like to kick us off, Steve.
I can also read them.
Yeah.
Hypometabolism is also causal for epilepsy. Ketosis alters energy productions and manifestation. Is this a reason? Yeah. So yes, one of the big reasons is the only way we know to diagnose epilepsy is due to seizures, and they can come from just like an inordinate amount of issues at the lower end. So the answer is yes. It's a lame answer because many things can cause it. Sorry about your audio. There are probably many ways audio could break in the same way, but we investigate all the same. Does bursting behavior change with alterations in ATP, NADPH, progesterone, or yeah, it changes a bunch. So Steve asked, so ATP, energy of the cell, great. Progesterone, IR, irradiation, neurotransmitter precursors. Those are all different interactions with the system, so they could for sure change it. A little bit of ATP is fine, but too much ATP is dangerous and will cause cells to die. And it's something we're working on right now. I think I can talk about it a little bit, where we can connect cultures together and have them communicate roughly. So like if culture 1 does this, culture B receives this, and that rule is fixed, so they can try to learn to communicate. And the idea is also connect this to like robotic perfusion systems, where like if this culture does this, dump this drug here, what happens in learning if these drugs are mixed in at different time scales, because they radically alter neuronal behavior in every case.
Audio just
Hey,
cooperated. So I'd like
nice.
to ask if you've considered either building a grid that is, in a sense, different sections of it are isolated from each other, where you could actually look at different influences of the responses, and whether or not you can compartmentalize, use a big grid, but compartmentalize the solutions in which these neurons are being grown and supported and developed, to see if the ripple pattern changes when it goes from one environment to another.
Yeah, that's a fantastic question. I think I have let me open up a slightly separate PowerPoint just to be able to show a picture, but I might not have it. We do have that, rather than waste time searching. So we have a system where we have, it's very difficult to, within one electrode array, to compartmentalize cultures completely because they all share media. Could try to build something, but it's very tricky, like in hands, because then the compartments are really small. So what we've done is there's a system, it's 6 cultures that are electrically connected. So they are not physically the same tissue. However, we can electronically have them interact. And then you could put a different media in one well, a different media in another one, a third one, a fourth one, a fifth one, a sixth one. And see, 1, just does the base behavior change? They were all plated roughly the same. And then 2, when they interact with these drugs, how do those interactions change? So yeah, it's on the docket. We've started some of that work, but some of that work is also a little expensive. So we wait for grants. It's a great question though. Membrane fluidity is easily modified by PUFAs. Yeah, they do. So basically I think the general theory of why cultures burst is everything has bioelectricity in it, right? So all of these things are like fats and salts with charges and ions moving around. So instead of thinking about, because I know when I was in the beginning, I was thinking about neurons as like these impenetrable units that did this one thing. But really, they're perforated salty sacs, and there's ions flowing around everywhere with sugars, and it's all going crazy. Um, so when enough neurons fire nearby together, you get these field effects. Sometimes this can increase communication between neighboring neurons through something called ephaptic coupling. So you could call it, very colloquially, like wireless communication. And so we think what's happening with bursting is that when enough of these kind of key neurons fire, the way that they're have naturally grown on the dish, they create a strong field, create the next set that fires, which then kind of cascades out. So anything that would alter any electrical property of the cell, whether it be the capacitive current when ions are flowing towards it, whether it be how excitable they are in general, will change how the bursting works. However, typically, I don't have the precision. There are engineering groups trying this where they try to lay the neurons in a very specific physical configuration. And they're trying to investigate when you do that, how can you start manipulating and understanding some of this stuff? But it is super duper hard. For me, I think at the end of the day, regardless of how you alter them, because I don't have the ability to alter single neurons yet, you would still see key groups that are responsible for certain burst dynamics. They're kind of starters because of the way their axons and cell bodies are laid out.
What kind of timeframes might be involved in like creating a gradient across one of your grids of some environmental influence, one kind or another?
So we can create an electrical gradient across the dish. So for example, you can align the stimulators in time and in space such that they go like pew. And so you can try to get artificial ionic waves going inside the dish. It's technically a little challenging. The timescales that are possible with that can be very fast. I think upwards of like one time I tested it and I got the stimulators going as fast as like 300 hertz, which is well within reason for brain activity. What's fun or interesting is whatever your interactions are, they need to be relevant to the system that you're looking at. So these unveiled signals, if you pulse too fast and neurons don't have time to integrate those signals into themselves, I don't think you'll see a lot of interesting phenomena. Conversely, if it's too slow, I doubt you'll see too much either. So there's kind of a Goldilocks region of how fast or slow the signals can fluctuate, where you're still going to see meaningful interaction. But like, seconds, sub-seconds is totally fine.
What about a chemical gradient where you might take the same pattern of cells and have an ATP gradient from one side to the other and then wash it out and have it go from the other side to see how the bursting activity might change?
You can do this. Fluids are very hard to work with. So I think the easiest way to do it is have an inlet on one side and an outlet on the other. You pump in a decent amount of ATP, and because this outlet will be pulling, you naturally will kind of pull the ATP across it. The tricky part of that is it's very difficult to control. So if you're trying to do systematic understanding of how a particular ATP wave or flow is traveling across your dish, it's just very hard. And then the moment a bubble appears, which is more common than anyone would ever want, everything that you've done goes a bit crazy because the bubbles are very catastrophic for flow. So it's possible. I don't want to do it. But if you know anyone that is good at it, I would love to have them come here and help me do it.
Thank you.
Micah, go for it.
Have you tried neurons from different organisms? Like we know, at least for humans, like the structure of the brain is different. Like we have the cortical columns and all that stuff. Have you tried fiddling with different animals or organisms who have different neural architectures or different structures and see if that yields different results?
Yeah. So I would say my first grant that let me do some of this provided enough funding for me to try with like one organism, but it's like the next thing we're going to do. I've tried with both mouse and rat, and They seem similar-ish, but my hypothesis is if you move into things like iPSC cells, they're going to be a little bit different. Those would come from humans. And then my next effort is trying to get. It's difficult because you have to move the machine between different environmental zones. So neurons like to be warm. Frogs don't. So you could take a frog brain, for example, because Mike's lab does a lot of work in Xenopus, and try the same thing in those cells. And then I just received a little bit of seed funding to work on planaria. So planaria, these infinitely regenerating flatworms, they have a brain. It's not small, but it's not big either. To see, one, if we can culture them and try the same thing, or lay their entire body down on the sensing array with their brain exposed and seeing if you can kind of train their brain inside their head. They're very weird. If you cut them in half, they'll regrow their head. So if you scrape off their skull, plop them down and do a thing and then leave them alone for a bit, it'll just regrow the skull. They don't have a skull, but they regrow the top of their head. So it's kind of a weird idea where can you like access their brain, train it, or do something, intervene, and then take it back off, and then it will still behave as a living full animal. That's probably the most buck wild thing that I'm trying to do. So yeah, it's something on the docket. So essentially, it's like the more funding, the better, because then we can grow new and fun things. Because I think being able to mix and match different phyla, different organisms, will help us kind of better understand some of these general design principles I spoke about. Hopefully that answered the question.
It did.
But do you have another one? I see a little icon.
I do. I was waiting for Lydia to give her
Go for it. Go for it.
Do you have thoughts on? So I found it very interesting, this idea that finding some sort of fundamental thing that neurons would like to avoid or are attracted to without relying on knowledge of the higher level biological organism, right? So their idea of they don't like randomness is the hypothesis, right? As they try to get away from it. Do you have other ideas on kind of more fundamental things, like some random biological system or even mechanical system or trainable system would like to avoid or is attracted to?
Yeah, no, so it's a great question, and it's, I think, the most received question in my life with this kind of work is like, what are good training signals and why? So just to be clear, the regular random thing, definitely not my idea. And I'm doing it, but I think formally comes from Carl Friston with the free energy principle. So in that mind that I'm way oversimplifying is that biology will always work towards regular because it's easier to predict. And ideally you would like to be able to predict and know so that you can act appropriately and not die. I think the not die piece is important. If you can find any signaling that is strong and without being overly destructive, I think is important. So in the case of the stimulation avoidance example, the one that Half and Synopian kind of put forward as how that's working is that when you kind of use voltage to perturb a system, especially at the strength that I did it, it's not volts and volts, that's too strong, but it's very strong with respect to the system itself. It's like 250 millivolts, keeping in mind that typical depolarizations are at like 70, so 200 is quite strong. And it's not constant, but it is, it's not that I keep it at that voltage, but it's coming constantly. So I think it's an extreme perturbation for the system, but does not destroy it. And I think that's a really strong one for a punishment. So basically, I think that's a very strong punishment signal for the same way that if every time you did someone slapped you really hard, like, yeah, it's uncomfortable, so you would work to avoid it, but it's also not going to destroy you. If someone shot you every time you did something, like you get one chance and it's over. So you can't be overly destructive. For a reward, I think Pavlov showed that tapping into any system like hunger is really powerful. I mean, I don't know if you could go find the book at a university library. He did some absolutely buck wild things where like he got the liver to do weird stuff by showing food to it. So he's tapping into some kind of deep system like hunger. So When I'm thinking about rewards for my system, for neurons, I'm thinking about giving them nicotine or other stimulants that could help. Punishment and reward work on different things. So hopefully that's helpful. So if you have a system that you want to investigate, it's sometimes not too difficult to find ways to punish it that's relevant to it. The reward one is harder. So, neurons, they can uptake nicotine and things like that. So, and there's already systems in the in neuronal systems that are in play that can handle that. So I think tapping into a system that's already there, like ATP, for example, could also be a good place to go.
Gotcha. Thanks. Just a quick follow on to that. Have you tried giving a stable electrical signal or wave as a reward? So just like you do the random one as a punishment, have you tried just giving some kind of, nice electrical signal as a reward?
Yeah, so not a constant one, but one of those graphs that I showed was when they did the thing I didn't want. It was the pulses came randomly, and I should have put it as a picture, so I apologize. So the punishment was a pulse that came at random intervals, and the reward was a pulse that came at very regular intervals, like bop, bop. One culture was perfectly trained by this. The other culture plummeted and did opposite of what I wanted. So I need to do more is the answer to that, but it's interesting. Yeah.
Were the pulses like square waves, or were they like harmonic sine wave pulses?
Easy peasy square waves, because the math was easier, as well as the technological approach. But doing sine wave pulses or coupling pulses together to create waves is something we're trying to do. We just need enough cultures and enough time to actually try it. That's the kind of hurdle now. It's just the time to do it all again.
Awesome. Well, we're at the hour. Thank you so much, Wesley. This is
Yeah, no
totally
problem.
fascinating. And it was great to see the rest of you. Thank you so much for joining, for your attention.
Yeah.
And we'll see you soon. Have a great
right.
rest of your day.